an breaking surprise news in medical science. Almost every human cell is powered by mitochondria, bean-shaped organelles that use oxygen to synthesize usable energy. These structures evolved billions of years ago from free-swimming bacteria that were engulfed by some of humanity's earliest ancestors. Because of this history, mitochondria still have their own ring-shaped DNA, completely separate from the 23rd chromosomes that makes up most of the human genome. And although those chromosomes come from both parents, nearly all humans inherit their mitochondrial DNA from their mother's egg cell. So what happens to the empty DNA in the sperm cell? Understanding this process is important for studying mitochondrial diseases, genetic disorders that result when these powerhouses don't function properly. Yeah, scientists know that molecular processes break down the sperm's mitochondria soon after fertilization in another animals. But no one has been able to pinpoint when this elimination happens in humans. New biologists have discovered that it happens early. In fact, human sperms' few mitochondria contain virtually no DNA at all. The findings were published in a recent issue of Nature Genetics. We were very surprised by the absence of empty DNA in mature human sperm because previous studies had produced conflicting results, says the study's senior author, molecular biologist Dmitry Tremyakov of Thomas Jefferson University in Philadelphia. This empty DNA elimination process might play a role in human infertility and can help science understand mitochondrial disease, he says. Using molecular biology and microscopy techniques, the researchers examined human sperm cells across their developmental stages. They showed that mitochondria in the sperm's precursor cells did contain DNA, along with an important protein called mitochondrial transcription factor, and also alphabetically A, that maintains and protects the DNA. Yeah, but when the sperm cells matured, a slight chemical change prevented TFAM from entering their mitochondrial. It entered the nucleus, where it could no longer prevent the empty DNA from degrading. If empty DNA sticks around its sperm's mitochondria, it could become a source of infertility. Previous studies showed that people with decreased sperm counts and motility have elevated amounts of empty DNA in those cells. Mouse studies have indicated that TFAM is absent in sperm's mitochondria, says Zihan Wang, a mitochondrial cell biologist at the Stanford University School of Medicine. The new study, along with prior research in other animals that found paternal empty DNA can be eliminated after egg fertilization, shows multiple mechanisms that may contribute to maternal mitochondrial inheritance in different organisms, she says. Tremyakov says there are probably other yet unidentified mechanisms that regulate empty DNA in different cells and that might contribute to mitochondrial disease if disrupted. We need to uncover this mechanism, he says, to better understand mitochondrial disease and how to treat them. Bien, report.